Hello, my name is Jenna Nelson, and today I'll be presenting my research on PFAS contamination in Maine agricultural areas. This is an assessment of sludge application sites for priority testing in the Little Androscoggin River watershed. Para and polyfluoroalkyl substances have been detected in Maine agricultural products, wildlife, and drinking water due to decades of industrial and municipal wastewater sludge application on farm fields. Hundreds of sites across the state remain to be tested. Therefore, a priority-based system is needed to identify sites that impact the greatest number of citizens and natural resources. This study identifies a process that can be used with available geospatial data to determine which sites, if contaminated, have the highest environmental impact. The processes were then applied to the 12 sludge application sites in the Little Androscoggin River watershed. Here's a close-up of the study area and the sites. Site boundaries were determined through historical records and satellite imagery. The boundaries were buffered to account for potential PFAS transport through the surface water or the vadose zone. Criteria for risk assessment included intersection with surface water features, aquifers, public water supply, private wells, and current cropland. These sites were identified as highest priority for testing due to potentially impacted private and public drinking water supplies. 3 additional sites may require additional areas for testing due to underlying sand or gravel aquifers. A significant amount of cropland throughout the watershed may be impacted due to its proximity to sludge application. Although outside of the scope of this analysis, all sites that are still used for food or animal feed production should have their products tested as soon as possible. For more information about PFAS and PFAS in Maine, you can pause the video to look at this introduction. But basically, I'll draw your attention to how starting in the 1970s, industrial byproduct sludge application permits were granted to hundreds of agricultural sites across Maine. The paper mill sludge was treated by municipal wastewater plants and then spread as fertilizer. Unfortunately, PFAS chemicals were present in some of the sludge, either from industrial waste or having entered the municipal stream through household use. Here we have some correlation between soil test results and known locations of sludge application. So here you can see that the color shows the concentration of PFAS in parts per trillion, a pretty big range. And we can also see that only some areas have been tested. Every single black dot represents a sludge application area. So there's a lot of testing remaining to be done. These soil data points have obviously been dispersed just to show the general central location of where they were gathered. According to the Maine PFAS Task Force, sites and materials in Maine that are under investigation for PFAS contamination include public water systems, groundwater, surface water, private water suppliers, retained milk supply, vegetation, corn and hay for dairy feed, sludge, and fish tissues. PFAS have been found permeating lands and waters near facilities where firefighting foams have been used, and where paper mill residuals were applied and spread on agricultural land. This study is going to focus on those sludge application sites. If you're interested to know more about different models for assessing possible spread through groundwater and soil, please refer to these studies that you can find in my references section. There are 12 areas with historical sludge application permits in the study area. These were on record with the Maine DEP, and these records were compiled from several sources, do need to be checked for accuracy according to the DEP. These farms obtained a license for application of municipal wastewater system treated sludge, but actual land application could vary. Again, because they spread the sludge, it does not mean it was necessarily contaminated with PFAS, but that is highly likely and is the reason why these sites need to be tested. The acreage of potential sludge spread and approximate coordinate points were determined from these permit records. Each of the points that were in the records were converted to polygons based on the acreage and satellite imagery of the fields. Historical imagery from USGS 1998 via Google Earth was viewed. In almost all cases, the boundaries of the farm fields have remained unchanged over the last 2 decades, so it's pretty easy to determine the likely spread areas. The perimeters of the farms are identifiable due to adjacent roads and forest. Due to the DEP points using these points as center points, polygons were created in the GIS to approximate the field boundaries according to the acreage recorded on the permits. Next, a buffer was applied to each polygon to account for potential pollution spread from surface application through the Vetus zone and into the groundwater. Each of the sludge fields were buffered 0.5 miles, and this distance has been used by Maine DEP as a guideline for testing private wells. This is what has been used in the Fairfield sludge investigation. And again, this data is from the Maine DEP. You can see something similar where they determined the field boundaries and then have basically warned everyone that all wells and water sources within 5 miles should be tested. However, this 0.5 mile buffer should not be considered a definitive boundary of potential spread. It could be a larger or smaller area. Really depends on how much was spread and different conditions of the ground. The EPA is still developing tools for modeling PFAS spread that will make specific site assessments more accurate in the future. So after I buffered those areas, I then took the buffered polygons and analyzed them for intersection with environmental features using the GIS. Locations of private wells, public water supplies, major aquifers, and surface water bodies, all data from Maine EGAD, were checked for intersection with the contamination zones. The Summarize Within tool was used to estimate the number of wells and acres of cropland potentially affected by the contamination, and the sites were then ranked. The highest number of impacted water features and acres of cropland were given the highest priority for preliminary testing. So here are my results. First, we'll look at which sites had the highest number of wells, and here they've been visualized with the darker red being the higher number of wells. And these are the specific ranges. The number of recorded private wells within the contamination buffer zone varies by site, but a few really stood out here. The Furland, McKay Torrey Farm, and the Witham sites have the largest number of private wells, each being 20 to 40 wells on record. There could possibly be more wells because this well database has not been regularly updated. Next, we'll look at potentially impacted aquifers. The Ford and Hemmond site buffers overlap, potentially increasing contamination of the aquifer underneath. Both sites intersect this aquifer below West Minot. The Brett site in Otisfield Gore also runs close to a major aquifer, just outside of that buffer. if that aquifer is impacted, that could extend the area that should be tested for PFAS. We also have several bodies of water that are potentially impacted. The Thurston site may have impacted Little Penisawasi Pond. The Furland and Witham sites are less than half a mile from the Androscoggin River, or the Little Androscoggin River, which then feeds into the Androscoggin River. And I also looked at cropland. Each of the buffered sites contained various acreage of current cropland. Again, I used the USDA raster. The 4 sites with the largest distribution of cropland each contain over 200 acres and should be prioritized for testing, especially if they're currently producing food for human or animal consumption. The largest cropland area is the Furland site with 359 acres. So that these are the largest site here and how many acres it has. And then I guess most of the sites were fairly small with about or slightly under 100 acres. So again, we have several impacted water features, especially at the Ford and Hemmond sites with the aquifer there. The McKay Torrey site may have impacted the aquifer below Rang Pond State Park. This aquifer even feeds a site for a bottling plant, so it's definitely worth investigating. But the Consumer Reports have found 1.66 parts per trillion in a sample of water, but it's hard to trace back exactly which site the water was bottled at or the source of the water. One public water system may be affected. It's just the Fairlawn Country Club because it's adjacent to the McKay Torrey application field. Using the buffer tool instead of a groundwater model has its limitations. Buffer tool should be considered more of an approximation than a model which considers the unique geology of the area. Darcy flow and particle track tools in ArcGIS would have been used in this study, but not enough data points were available from USGS for interpolating a groundwater elevation raster for the study area. There's still much research needed to determine how accurate those tools would predict PFAS spread in the Vados layer. In conclusion, my priority testing recommendations would be for Furland, McKay Torrey, and the Witham sites. Due to the number of private wells, acreage of cropland, and proximity to the Little Androscoggin. Fish samples should also be taken from the Little Androscoggin as well. The Brett, Hammond, and Ford sites should be tested due to the number of wells and proximity to aquifers. If PFAS levels are high, the testing should extend to wells impacted by those adjacent aquifers. Again, these are just suggested contamination buffer zones, and this is just recommendations for testing, not saying that these particular sites are contaminated, but sludge was likely spread in those areas, and there's definitely need to test all of these areas. Thank you for watching, and feel free to consult my references for more information.